China. So the initial news in December 2019, when we heard about an unknown pneumonia outbreak in mainland China, some of us got worried that this could potentially be a new respiratory pathogen. So at the time, I was a postdoc at McMaster University, and I reached out to my colleague at the University of Toronto. And we had a team meeting and decided if the virus ever came to Canada, we would get samples, we would isolate it, and we would rapidly characterize it because that could help vaccine development and diagnostics and research. And unfortunately, the virus did arrive in Canada about last week of February in 2020. And when the virus did arrive, we sampled the first three patients that were diagnosed as COVID-19 positive. So we collected these empty swabs. These are swabs that go up your nose. Some of you folks may have gotten these tests done. And at the time, this was very early days. The WHO had not called it a pandemic yet. This was just starting to look like an epidemic and eventually turned into a pandemic. So we took these samples from this clinical specimen from patients. We put them on Virome E6 cells. Now, these are immunocompromised monkey kidney cells. And we were able to isolate the virus in these cells, which we then confirmed using qPCR. So most of you know what a qPCR is. This is what's being used for diagnostics. And we also sequenced the whole genome of the virus using nanopore sequencing and mini-seq sequencing. And this was the first image of the virus isolated in Canada. And let's walk you through this. These are human lung epithelial cells, and where you see all of these electron-dense particles. So this is an electron micrograph. We use an electron microscope to do this. And all of these electron-dense particles are SARS coronavirus 2 particles. And if you zoom into one of these, I hope you can appreciate this light gray halo outside of the virus particle. So the virus gets its name coronavirus because corona means a crown. The spike protein forms a crown around this round virus particle. And this light gray structures on the outside is the crown that this virus is wearing. So we knew we had